Yeah, it's like anyone for like
well, yeah, that's also the new one.
oh, I know. I need to get off of that one.
Like Miranda, she does that too.
You have the seminar every week?
I just I missed the shit.
Yeah.
It's quite, like we have a good budget for it. So.
Oh, thank you.
Yeah.
There's a clock there also.
Yeah, I saw that. But usually you want to be done by the full hour? Is that how it works? No,
Why?
they don't kick us out.
No.
There's no class after
No.
the rest, so okay.
Just confirming the stream is on, the audio is working. We had some trouble with the audio yesterday, but this looks all good.
Question.
Yeah.
How long do you be available?
Oh, totally, yeah. I can, it's gotten to this default state where it flips to unlisted.
Yeah.
So it's still there, but it's kind of security through obscurity. But if we want to have it visible when somebody clicks on that link, I can just leave it
Or you can just
public.
Or
rather
just
than
it doesn't matter.
okay, I'll send it to you when it's done. Yeah.
Yeah.
I think.
Whatever's
Yeah.
easiest to.
So there was a talk in here at 9:30 from EEB. So that they were in here, and sometimes the ingester at YouTube, it just smushes them together,
Yeah.
but hopefully it's not. I think I made an explicit cut about 10 minutes ago, so we should see that separate. Yeah. Okay. Thanks.
Thank you.
It's on you.
Yeah. He gave a talk in Chicago 2 weeks ago, and then halfway through it's also like zoomed. Halfway through, Zoom broke down.
Ours works. It works pretty good. I don't think we have too many issues with it. We share our IT with the EB department, which is mostly in another building.
Okay.
They
Do you work much with that department too then?
Like, not as much as I'd like. It's a little bit more separate, I think, than it could. We have like a couple cross-appointed, but they're not really. Like only really Belinda, like we meet with this afternoon.
Yeah.
She's like truly like financially cross-appointed.
Yeah.
Everybody else is just like in,
In one or the other. Yeah. Yeah, I met her. When was it? 2019. We had a conference at MSU that I co-organized, and she was one of the speakers. Linda was, yeah.
She was saying the skull was like so soft.
Yeah, there's a couple people over there. But, I think like there's also good communities at the satellite campuses. In there's still like single departments. So it used to be zoology and botany. And then they were merged and then split to
okay.
and South
Yeah.
But in the satellite campuses, there's still kind of one department.
Okay.
And so there's a lot more, people that do both.
Right.
I don't see it.
Yeah, I find I'm fine. Things differently.
Like we had a choose a person. I think they sort of finished.
I was like so upset, so you like the whole time. It was, yeah, I know.
Which is probably usually.
Once I sat down, like 5 minutes in, I was like,
people that listen in, do they also ask questions? Or is there no way to?
I don't know that some people listen.
Okay.
I think usually they come in person. But there's no mechanism. It's just that
Okay, it's not like it's you. Oh, okay.
Yeah.
It's just streaming it out. Yeah.
I think in EEB they sometimes do like a Zoom. But most people are in this building, right? So then it's just they just come in.
Yeah.
So you were surprised?
Hans Hoffman. I had a meeting with him. I might end up going down there for a talk. I had like a Zoom meeting with him a while back.
Yeah.
I asked for.
I mean, I don't know him well. When I was in Malta, or with Axel, the first microbrewery was built with Hans.
Right, yeah, he was saying.
So then Walter went to Harvard when Hans was our fellow at Harvard.
Yeah, he was telling me about this.
And then I did the sequencing.
Oh, yeah?
I was the person that did the sequencing for the EST library to figure out what's actually on the chip.
You were telling me about some meeting you had with Eric Lander for the genomes and stuff like this, and how like Eric Lander's like was double booked, like all his meetings were double booked, and he was like going back and forth between two rooms or something like this. Like they started the meeting and then halfway through he came in and like said something, and then he went back to the other room and there was the rest separated by glass or something.
When I was at the Broad Institute for the coelacanth and guar genome meeting, or it was just coelacanth. It was a very intense meeting and so many people, and everybody of course wanted to know everything. And I think that in the last final discussion he had snuck in and sat in the back with his bare feet like on a table, and I think there was some heated discussion in the front, and then he was like, guys, I think the coelacanth is the coolest since the mouse genome, and you should just all work together. No, he became Biden's science advisor, and then there were some problems brought up about like how he treats his employees, and then he got fired basically, or he resigned from their job. And I don't know if he went back to the MIT Broad or.
What do you do now? You don't need this czar to tell you which genome to sequence anymore. Now you just sequence the genome. What does
Oh yeah.
That's all.
It's all right. Sure.
Okay, I think we can get started so that we have enough time. So it's my pleasure today to introduce Ingo to give a talk here on fish genomics and comparative genomics. So Ingo did his studies, started his career in Germany, where he's from, so at the University of Constance first, and there he worked a little bit with cichlids, but then I guess turned away from the light of cichlids and went to other fish. For his PhD, he did color pattern stuff across fish, and then for his postdoc, he joined John Postlewaite's lab in Oregon, where he started working on these sort of holostone, living fossil fish like the gar and things like that. He's been a really big component of sequencing these genomes and annotating them, and doing some really cool stuff with them, not only for the GARD, but other genomes as well. And I guess today we're going to learn a little bit about gene regulation, but I think his lab does a lot of different things. That's really integrative, some neurobiology, some developmental biology, ecology, and mostly a lot of genomics. So let's welcome Ingo and hopefully have a nice time.
Yeah, good morning everyone, and thanks Max for the invitation. It's great to be here, and I look forward to meet many of you over the course of the day. So my lab at Michigan State is the Fish Evo Devo Geno Lab, and I'm in the Department of Integrative Biology, and our motto is understand life in context from molecules to ecosystems. And along that trajectory, my lab is really involved in the understanding the relation of genotype to phenotype, and then how the evolution of the genome and phenotypes lead to biodiversification. And I like to do this in fish because they are a fantastic model system to understand a lot of biodiversity at the genomic level, at the phenotypic level, to be able to link these connections between the genome and the phenome. And I also like to look at this through the lens of development. So this is the first day in life of a zebrafish embryo. You see the yolk here, cell divisions happening here on top, and we get this ball of cells. Now we go into epiboly, where the cells move across the yolk and invert, and within less than 24 hours we get a pretty well-developed zebrafish larvae with a head, with an eye and an ear will come up here, then the trunk with the somites and the tail. So after less than 24 hours we have already a little vertebrate embryo, and you can do that in fish pretty easily. You get hundreds of eggs. They're transparent very often, and it's just a beautiful process that you can look at over and over again. So my lab is basically doing some comparative genomics, but also Evo-Devo studies in fish to link the genome and the phenotype. And we do this at a more macroevolutionary scale, and are very interested in major transitions across the evolution of vertebrates, all from the dawn of vertebrates to the more derived lineages within the bony vertebrates. And we would like to understand how genome evolution in all its facets, like the structure of the karyotypes and chromosomes, the genes that they encode and the way they are regulated, how those then at the macroevolutionary scale have contributed to morphological evolution and to many of the morphological innovations that lead to these clade-defining traits within the vertebrates. We're mostly focusing here on the bony vertebrates with the two main lineages, the ray fin fishes and the lobe fin fishes, which then lead to the tetrapods and ourselves. And the research in the lab can be broken down in three main branches. So one is we want to understand the biodiversity of the teleost fishes. So the teleost fishes within the ray fin fishes are the largest group of all living vertebrates, more than 30,000 species, about 50% of all vertebrates. And we would like to understand how this biodiversity came about, specifically in the view of a whole genome duplication that occurred in the last common ancestor of all living teleost fishes. And I will get back to that towards the end of my talk. And then secondly, we are also very interested to learn more about ourselves. So how did tetrapods and the human genome and the human biology came about from fish ancestors? So what are the genomic and morphological transitions that happened when fish crawled on land and became tetrapods? Because we are, in a very strict sense, we are just the tetrapod fishes, right? So we are a special type of fish that evolved the capability to live on land. And then when we put this together in a more applied way, then this will also help biomedical questions, because the development of fish and human are actually quite similar if you look at it. So this little embryo that we generated earlier in less than 24 hours resembles very much like a less than a month-old human fetus. And we see a lot of similarities in the developmental processes. And of course, the zebrafish and other teleost fishes are popular biomedical model systems. So we have zebrafish as the most popular one. We have the Medaka, which is also a generalizable model that is coming out of Japan. The platyfish that I worked on during my PhD is an established classic cancer model for melanoma formation. We have the cavefish with interesting phenotypes concerning eye loss, obesity, and metabolic syndromes. The ice fishes, which have reduced bone to lift them up in the water column and are a model for osteoporosis. The killifish has become a very popular model recently for aging. And then we have some more evolutionary-based models like the electric fishes for communication, stickleback for a lot of ecological adaptations, and of course Max's cichlids with all their diversity and phenotypes like sleep patterns and so on. So if we want to help the community, the zebrafish and the Telios model system community, a lot that we can learn from our evolutionary comparisons within bony vertebrates should also inform biomedical questions. So this is really what we are set up for then, this what I call the T2T, Telios to tetrapod comparison. And what we want to do is basically reconstruct the bony vertebrate ancestor, because we need to figure out how the bony vertebrate ancestor looked like to then make assessments how evolutionary divergence in these rather derived lineages came about. And of course, we cannot go back in time and then sample a species that's close to the bony vertebrate ancestors like Sarolepis here and sequence its genome. So we are thrown back at the extant lineages and need to reconstruct the bony vertebrate ancestor from there. And then figure out the content of the bony vertebrate ancestor and how its morphology looked like and what novelties and losses have occurred independently in the lineages leading to Telios and tetrapods. And these comparisons are complicated by the fact that we have 3 rounds of whole genome duplication or polyploidization that we need to take into account. So before the bony vertebrate ancestor, we had 2 rounds of whole genome duplication, round 1, round 2, that led to basically the amplification twice of the genome and its genome content, and then the majority of extra copies you tend to get lost, but in lineage-specific manners, so that then the components of the genomes between lobefin fishes and rayfin fishes will differ sometimes quite drastically. And then we had the extra Telios whole genome duplication, the ancestor of Telios, which then further complicates this comparison because very often we look at 2 genes in comparison to human and other tetrapods. So this is kind of the setup that we are looking at with all these innovations at the genomic level, at the phenotypic level. And we try to set out, when I started my postdoc and thinking about what can we add to the comparison. At the time we had tetrapod genomes, we had zebrafish, medaka genomes, but not really much. And like when we want to study, for example, the Telios genome duplication, tetrapods are not really that great of an outgroup to them. So we were looking for establishing new fish models at the genomic and developmental level. So a fish system that would have diverged before this Telios whole genome duplication, that should have an informative genome, so not very derived in its own way. And then because we want to do evo-devo studies, also available for embryological studies. Now, you could think like, what about the lobefin fishes, like the lungfishes and the coelacanths? They are, of course, a first guess for studying the lobefin branch more. And we were involved in the coelacanth genome project, but the coelacanth is probably the worst model system you can ever think of. It's highly endangered, it's deep sea, it has internal fertilization, so nothing is going to happen there beyond the genomic level. And the lungfishes have genome sizes that are 30 times the human genome, like 90 gigabases. So genomes are only now becoming available in a very fragmentary way still. So besides some comparative genomics, we are kind of not going there. So what we decided is to look at the so-called holostean fishes, the gars and the bowfins. And as I will tell you, so the holosteans have a key phylogenetic position So they are within the ray-fin fishes, they're the closest living relatives to the teleus and to the teleus genome duplication. And thereby they diverged before the teleus genome duplication, although at the time when we started this was still a matter of discussion. We found that they have highly informative genome structures and very slowly evolving genome sequences and also slowly evolving body plans. And we can do comparative developmental biology with them. So, the cool thing about the holosteans is that they have been around for a very long time in a very inert, evolutionary inert manner. So they are sometimes called the ancient fishes. They literally swam already with the dinosaurs. This is here paleo artist rendering from the mammalian megafauna. And the gar and the bowfin that are shown here pretty much resemble the fish that swim around today. There's only 7 to 9 species of the holosteans compared to 35,000 species of teleus. But that's a little bit of an illusion. So when people had looked into the fossil record, the holosteans were way more diverse in the past, and the diversity kind of matched the diversity of the early teleus fishes. We're kind of in a lucky situation, so to speak, because the bowfin and the gar diverged very early during holostean evolution. So by comparing the two clades, we should learn a lot about the remaining genomic and morphological biodiversity within the holosteans. And they, the holosteans have these archaic body plans. So when we think of the bowfin, its skull is used in comparative vertebrate anatomy labs as being the prototypic ray-finned cranium before the innovations that happened within the teleus cranium. The gar has these ganoid scales, which are hard-coated scales. Basically what we show, it's kind of similar to enamel on our teeth, and it's made by similar genetic programs, which is also the ancestral condition for bony vertebrates. And then both of them have respiratory gas bladders, so they come to the surface, they gulp air, and that helps them to breathe in addition to breathing with their gills. And these are all kind of ancestral conditions that were also found in the bony vertebrate ancestor. So we had to choose between the two. Where would we start? And we actually started with the gars. And what are the gars? So the gars have been around, as I said, for a long time, more than 100 million years. And already Darwin recognized that they are this very old clade. He called them the ganoid fishes because of the ganoid scales. And when he coined the term living fossil in The Origin of Species, he used the gars as one of his prime examples for the term living fossil, meaning that they're species poor, but importantly also that the living species very much resemble the extant living species. There are two genera of gars, Lepisosteus and Atractosteus, and a total of 7 species. And then we also had to make another choice, like which species to work with. And of course, you don't want to do this in the laboratory. So this is the alligator gar. It's one of the largest and fastest growing freshwater fishes. They are really cool. And we actually indeed try to work now in hindsight with them, because they are also aquafarmed to some degree for restocking purposes. But we decided that we need to work with the spotted gar, which was one of the smallest species that we can manage in the lab. And we work with Alice Ferrara at Nicholls State University in Louisiana. It's like one hour out of New Orleans, and she's a world expert in the reproductive biology of gars. So we go out in the field, we get, with electrofishing, we get broodstock, and then we inject the gars with ovoprim hormone, which leads to the release of gametes. And here you see a larger female with a smaller male in their mating process. So she releases eggs, he fertilizes the eggs, and then the eggs are actually put down on this artificial seagrass, which is literally pom-poms from MSU cheerleading. And then we can scrape off these eggs from the material, and then we can they're really hardy, and we have worked out nicely how we can grow them up to adulthood now, and we can of course then do developmental comparisons and do that in comparison to zebrafish and mouse and whatever we want to compare it to. So this is a collective team. So we are here at Michigan State. This is the distribution of spotted gar. We have this peripheral population, but they are really sparse there. So we actually go to Nichols State, and then Elise and Quentin have all the gear to go out into the bayous, and then we can fish for them. We can ship the eggs to MSU. That's the collective team that built the capacities and facilities up there at my university, and we are now also in the process of getting our first in-house spawns, so that we don't necessarily always have to rely on wild-caught broodstock. So at MSU we have established what we call the Ancient Fish Facility with the help of an NSF Edge grant that is providing funding to new model organisms for functional genomics. And this facility is meant to be a hub for the vertebrate research community, so we can provide live fish samples, protocols, and also house guest researchers to do their experiments. So we keep about 400 of these fish right now, but we need to stop adding more. So when I did my postdoc in Oregon, there was no genomic sequencing initiative yet. I started in 2010, so that was not where genome assemblies were really in reach yet for most non-traditional model organisms. So I started sequencing in John Postlewaite's lab some bug clones, but then one day the Broad Institute called and said, hey, we have sequenced the spotted gar for you, because John had submitted a white paper a few weeks before, so I threw all that away after 2 years of work because we had the genome. And it was based on a like Schwadron Illumina sequencing hooked onto a genetic map that John Flatt had generated at the same time. So it was one of the first non-traditional model system chromosome level genome assemblies. And that's the one that's still in the browsers, like in Ensemble. So if you're interested, you can go there. We were very happy with the quality. Over 90% of the assembly were on these 29 linkage groups or chromosomes. And then when I started my own lab in Michigan, together with my first postdoc, Andrew Thompson, we then also started to establish the bovine genome sequence. This time using high-C contact mapping to put the scaffolds onto chromosomes, so we got a nice chromosome level assembly for the bovine out of this. So that we have the comparative value of both Holostein lineages being covered. But of course, genome sequencing is a rapidly evolving field, so now we are working with the Vertebrate Genomes Project, which is currently just wrapping up phase 1 with the goal of sequencing at least one representative of all vertebrate orders. And bovine and spotted gar are included in there as representatives of the Holosteins. And now we have haplotype resolved genome assemblies for both species that are near gap-free and are of the highest quality. They're not telomere to telomere yet, but we're getting there soon, I think, as well, with another iteration of this. And they're now part of this whole initiative covering over 500 species of vertebrates. So I would like to give you some vignettes about what we can learn then from the Holostein model system. Reconstructing the ancestral vertebrate genome, and then also how they can be very informative to study gene regulatory landscapes in vertebrates. I'll talk a little bit about our most recent efforts to look at the evolution after whole genome duplication in the teleos fishes with the Holosteins as an outgroup, and then what other avenues we are taking to make the gars a more functional laboratory model to do some real functional genomics in them. So let's start with the reconstruction of the ancestral vertebrate genomes. So here's the comparison of the spotted gar genome. So this is still based on the old genome, but it's basically nothing has really changed about this comparison, at least. So we are comparing here in the circus plot the 29 chromosomes of the spotted gar to the 24 chromosomes of the medaka, which is a teleos fish. And then these lines and ribbons connect the orthologous genes, homologous genes between two species, both species. And when you look at it like this, it looks very confusing, but when you zoom into individual chromosomes, you see that there's a 1 to 2 double conserved synteny pattern. So typically one chromosome in GAR is homologous to 2 chromosomes in a teleos fish, which of course is exactly what you would expect by a comparison before and after the teleos whole genome duplication. So when we did this, this was a final evidence that the spotted gar hadn't undergone this same genome duplication. And then our real surprise came when we did the comparison of the gar and avian genomes, or the chicken genome, because it is a very clean comparison where you see a lot of one-to-one conserved synteny, especially in the smaller chromosomes. So we see extensive one-to-one conserved synteny between gar and bird karyotypes, which we, for example, don't see when we do comparisons of gar and human. And this is due to the fact that both the avian genome and also the gar genome is organized into macrochromosomes, so large chromosomes, and then these dot-like smaller microchromosomes that are by definition smaller than 20 megabases in size. And it's especially those small chromosomes that show this extensive one-to-one conserved synteny, which tells us that the microchromosomes were a component of the bony vertebrate ancestors genome. And when we include then other genomic information like the bowfin and teleosts and mammals into account, basically this was the culmination of a lot of comparative genomic work to reconstruct that the bony vertebrate ancestor had macro and micro chromosomes, which we still then find in the gar genome, but also in the birds, and also in other reptilian lineages like snakes or turtles and so on. And then other lineages have undergone mostly chromosome fusions, but also chromosome extensions by repetitive elements to get rid of these microchromosomes. So the bowfin itself actually doesn't have microchromosomes. There were some chromosome fusions that happened after the divergence of the gar and the bowfin lineage. So the bowfin is not as representative of the bony vertebrate ancestors genome. In the teleos lineage, we first had chromosome fusions, which were independent of those or different from those in the bowfin, and then the genome went through the teleos whole genome duplication, and thereby we end up only with macrochromosomes in zebrafish and other teleosts. And then in the lineage leading to mammals, we have chromosome fusions and other genomic rearrangements that also led to the vanishing of microchromosomes in our own genome. So what this all tells us is that the holostean fishes, but especially the spotted gar, has a very informative genome organization coming from the bony vertebrate ancestor. It is probably within the bony vertebrates, it's the best representation, or the closest proxy that we have as a genome to the bony vertebrate ancestor. Something we didn't look out for, and we were very fortuitous that it shook out that way. What about gene regulation now? What can we learn about gene regulation by having the holostean genomes in the mix? So for those that work with zebrafish, very often in a biomedical setting, one challenging observation has been when you want to find sequence conserved regions, especially non-coding space, which are then potentially gene regulatory elements, very often you don't find any sequence conservation if you compare human mouse and, let's say, zebrafish. So gene regulatory elements that are known from tetrapods don't show up in the teleos, and gene regulatory elements known for teleos very often don't show up as being conserved to the tetrapods. One confounding factor that is really important here is that the molecular clock of sequence evolution in the teleos fishes ticks very fast, potentially also as an outcome of the teleos whole genome duplication. What we and then others have found is that the clock in the holosteans ticks very, very slow. So we saw that with the first genomes from Gar and Bofin, and then this is a follow-up study by Tom Neer's lab, where they actually could show that the Gars have the slowest rate of sequence evolution within the jawed vertebrates. The Teleosts have the fastest. And then others are somewhere in between. The closest we get to Gar are crocodilians and turtles within the tetrapods. So holosteans have the slowest evolving genes. This is all based on gene sequences. By extrapolation, we think genome sequences. So how can we make this to our advantage? So we proposed the holostean bridge approach, because then the holosteans have a very slow rate of sequence evolution. So to travel in sequence divergence, so to speak, from human to Gar is shorter than from human to zebrafish or from zebrafish to human. You should go through Gar. So basically, the principle here is you align sequences from tetrapods and human to Gar, from zebrafish and other teleosts to Gar, and if these alignment blocks overlap in the Gar genome, although they don't seem to have sequence conservation between teleosts and tetrapods, we can indirectly infer that they are orthologous and they came from the last common ancestor of bony vertebrates. We've done this with whole genome alignments at the gene level. So this is from the early days, where we looked at, because we have this interest in the fin to limb transition during the water to land transition, where we looked at the data from the Vista Enhancer browser for human and mouse sequences that were tested in mouse reporter assays to drive expression into the developing limb bud in mice. And looking at our whole genome alignments, we could then assign a large portion of these elements back to the bony vertebrate ancestor, because they were conserved in the Gar, or we could find sequence conservation in Gar where zebrafish and other teleosts failed. So by that we could debunk some ideas that these elements were actually innovations within the tetrapods and already there before the water to land transition. And this extends beyond sequence conservation. So when Gar elements are tested in a reporter assay in mouse here, this is from the HOXD cluster, the Gar element drives expression into the developing limb bud in mouse, basically undistinguishable from the endogenous mouse element. And when we take that into the zebrafish reporter assay with a pectoral fin here, a similar pattern emerges where the Gar element drives as well as the zebrafish element. And we were very happy to see that others kind of followed in our footsteps here. So Mike and Ian here at SickKids took a similar approach by transitioning sequence conservation of open chromatin regions through the Gar system with indirect alignments to find a higher proportion of elements, gene regulatory elements that are conserved between human and zebrafish, to then do their follow-up analyses in zebrafish to look at enhancer elements that are important for heart development. Here's a more recent example that we've worked out. So one interesting feature of chordates, so our phylum, is the notochord, which is the namesake of our phylum of chordates, right? It's a stiffening structure and that runs along the anterior-posterior axis, and that is, of course, embryonically present in ourselves, and then later on in mammals becomes the intervertebral discs. And we are interested in how did the notochord come about, like a hallmark of the chordate. So by understanding the development of the notochord, we will understand not only how chordates came about, but it also has important biomedical relevance because cancers of the notochord, chordoma, so cancers of the neck and spine, are also based on malformation of the notochord. And an important regulator of notochord formation is the brachyury T or TBX-TB gene, so the T-box transcription factor. At first, and so to learn more about the connection of the development of the notochord, but also how chordomas can form, we first had to figure out what we're actually talking about when it comes to the brachyury gene. Because there's two brachyury genes or TBXT genes in vertebrates as a result of the second round of vertebrate whole genome duplication, TBXTA and TBXTB. They're both in GAR expressed in the notochord. The TBXTA gene is the famous no-tail gene in zebrafish, so if you have a knockout, you get a no-tail, as the name implies. But the TBXTA gene is actually lost in mammals. So in mammals, we only have the TBXTB gene. So we focused our first analyses on the TBXTP gene. And we collaborated for this with Alexa Burger and Christian Nosiman at CU Anschutz in Colorado. What they had done from a more biomedical perspective, looked at patients with chordoma and looked at chromosomal copy number variations around the TBXTB gene, which is shown here, and overlaid that with ataxic peaks from the notochord and some ChIP-seq data for TBXTB itself, because TBXTB is known to regulate its own transcription in an autoregulatory manner, and identified a couple of candidate regions which were only conserved to opossums, so within mammals. But nevertheless, they started testing them in zebrafish and found three elements that drive the expression into the developing notochord in zebrafish: T3, C, and I. And then Spinner Cosmic in Prague did a serial deletion of these three elements in mouse. And when all three elements are deleted in mouse, then we completely abandon notochord formation and TBXTB expression in the mouse. There's only expression in the tail bud, which seems to be regulated independently from these three regulatory elements. But then the question was, like, where did these elements come from? So there's no obvious sequence conservation to teleost or zebrafish. This is when we then applied our holocene bridge approach. So this is what had been found before, so only sequence conservation within the mammals, no obvious sequence conservation within the tetrapods, or not even thinking about fish. But we actually found that all three elements are in the spotted GAR genome, and from there we could establish them, two at least of them, in the teleost fishes and also in the coelacanth. And they are actually exactly where you would expect them with regard to the location of the brachyury gene in all three lineages. And then once those elements are tested, T3 from GAR or T3 from zebrafish, and the same applies to element I, they drive expression into the developing notochord either in a transient manner or when we have a stable line into the developing notochord. So there's also functional conservation here. And what about the tetrapods? I told you that the turtles have a slow rate of molecular sequence evolution, so we reasoned maybe they are also good bridge species, and that also turned out to be the case. So now we could find the elements all across tetrapods. Could even find them in the cartilaginous fishes, so rays and shark, but nothing in the jawless vertebrates, lampreys and hagfish. Which leads us to propose that this regulatory program comes at least from the jawed vertebrate ancestor. And when we look at the elements in more close detail, we found that especially those regions that show the highest sequence conservation are actually the T-box binding sites, the T-box elements. So suggesting that this regulatory program is derived from the jawed vertebrate ancestor. We haven't been able yet to figure it out for TBXTA and then maybe what's shared among the two, but that's something we're currently looking into. So this was mostly based on sequence conservation, but what about other means to identify gene regulatory elements? So this is something as a component of what we call the Project GAR code that we are trying to launch and find funding for. So that's our kind of mini encode now for the GAR, because we think it's so instrumental to learn more about gene regulatory programs across vertebrates. So we have these new genome assemblies, whole genome alignments, And we have done a first screen for open chromatin regions through GAR development with a TAC-seq. That's spearheaded by my former postdoc Andrew Thompson, who has now his own lab at Western Michigan, and graduate student Tony Zhu. We're also getting more and more into chromatin architecture to look at topological associated domains and loops within the TADs to figure out what's going on there, and we also want to push that to the single cell level. So we see some nice progression as expected through the developmental program at the level of RNA-seq expression in GAR and also at the level of a TAC-seq peak. So that's ongoing right now to figure out what these elements are actually doing. Here we have some first glimpses of how this will all shake out. This is actually from the bovine analysis, where we also had the chance to get some embryos. We have some high-C data to look into the TADs, and for a couple of stages, a TAC-seq data. This is just one example here from a TAD that contains the hemoglobin gene cluster, which is scintillally conserved across invertebrates, where we found open chromatin regions in the bovine in the surrounding of hemoglobin genes, which is the first time to show that these regions are conserved and open in fish, because these elements are known to regulate actually the hemoglobin gene expression. So again, we see some deeper homology of gene regulatory elements when we include these living fossil fishes. So we found about 140,000 non-coding open chromatin regions in the bovine. 50% or so are conserved with GAR, and about a little less than 4,000 are conserved at least by sequence conservation across bony vertebrates. But we are now doing more extensive alignments, and we're pretty sure that this number will go up the more species we integrate. And interesting, when we look at the Vista Enhancer browser data elements again, more than 55% of those elements that are show regulatory potential in the mammalian system are non-coding open chromatin regions also in the bovine, suggesting that there's a very old program at play. Here's some examples around some genes that we pulled out that have been previously studied to be regulatory elements for brain development, heart development, and fin and limb development. So those are all open chromatin regions in the holostein system. One is particularly interesting, the TBX4 so-called lung enhancer. It's an intronic enhancer in the TBX4 gene, which is important to orchestrate the early development of the terrestrial lung in mice. It couldn't be found in teleos before, but we now, through the GAR bridge or through the holostein bridge, we have found it in the teleos fishes, and it's open at the exact time point when the gas bladder forms in the holostein fishes, and also when TBX4 is expressed. So we think this is an old program suggesting that these air-filled organs, terrestrial lungs, gas bladders in holostein fishes, and also the swim bladders in teleos fishes, all come from the same original source. That's a question that has been debated since the days of Darwin and Richard Owen. Okay. Some words about the evolution after a whole genome duplication. So, as I told you, we have about 30,000 species of teleos, all derived from this ancestor that was tetraploid. And then we had rediploidization, gene losses, but then in the modern genomes we find a fraction of about 20 to 25% of genes being in 2 copies in teleos. And these are not always the same 2 copies. Like when you take a ciglet, you find certain genes in 2 copies and others in singleton, compared to GAR. And when you then compare that to zebrafish, you find a different subset of genes. So some of the genes that will be in 2 copies in ciglet will be just 1 copy in zebrafish, and vice versa. So although the number at first seems rather low, there's a lot of potential for diversification by differential retention and loss of these genes within the teleos. And then integrating more and more genomes and looking at what types of genes have been retained higher than the genome-wide average, we found that very strikingly genes that play a role in the central nervous system are over-retained in 2 copies after the teleus whole genome duplication, with retention rates of more than 50%. So we reconstructed that some earlier data, the patterns of retention and loss of these extra copies. So here we had the teleus whole genome duplication, and 1 minute after the teleus whole genome duplication, these duplicates were in 100% retention. And then we can reconstruct the decay or the loss of these genes over time. Oops. And you see as a first observation that most genes get kicked out right away after the genome duplication in a few tens of millions of years, but then already differences in functional groups established in their retention rate. So we have liver genes that are retained around the same level as the genome-wide average, so nothing special about it. During my PhD, I worked a lot on pigmentation and found that we have a retention rate at about 35%, which was already significantly higher than the genome-wide average. And then genes that play a role in neuronal differentiation and neuronal functions have retention rates of 50 or more percent. There's some striking outliers like retention differs by gene function. There's some striking outliers like the glutamate receptors, which have a retention rate of about 75%. So almost all genes are coming in 2 copies in the teleus fishes. And this is not only happening after the teleus whole genome duplication, so the sturgeons and the paddlefish, the acipensary forms, which diverged earlier in ravenfish evolution, they have gone through their own genome duplications, polyploidizations. GAR again is our outgroup, so we have a nice 1 to 2 conserved synteny to the sterlet, which only went through this first round of sturgeon genome duplication. So we also wanted to know what's going on after that genome duplication. So here we can see we have this high retention of all these glutamate receptors in the teleus after the teleus genome duplication. So human genes are just 1 block, likewise in GAR. And then if you see two blocks, that means both are retained. We have very few losses within the teleus. And then we look in sterlet sturgeon, and we see that almost everything is also then in 2 copies, which is quite striking. Like almost 90%. They have an overall higher retention rate anyways, but it's significantly higher for neuron genes in this genome duplication. So what's going on with these extra gene copies? I'm not a neuroscientist by training, but I closely collaborate with the lab of Julia Ganz, who is a comparative neuroanatomist, and that gives me the opportunity to look into this in more detail. So we take the GAR brain, which is a kind of our proxy to the pre-teleus genome duplication ancestor, and we compare that to the zebrafish and other teleus. And then we look at gene expression patterns through the brain, look at their expression domains, and then we might be able to see regions where we have 2 regions in GAR that show expression, where then 1 copy in zebrafish has lost the expression domain and the other copy has lost the reciprocal one, which would be an indication of subfunctionalization in the teleus brain, or potential specialization in the teleus brain. Or we might see regions that are expressed now in zebrafish that the GAR doesn't show, so that could be indication of neofunctionalization, so a new expression domain that emerged after the genome duplication. Another mechanism how the teleus fishes make use of these extra gene copies in novel ways, and thereby change maybe their brain morphology or their brain functionalities. So this is work from my graduate student Emilia Lorena and some fantastic undergrads. They did this atlas of glutamate receptor gene expression in the GAR by in situ hybridization for these glutamate receptor genes, and then compare that to the zebrafish, which is of course a massive undertaking, also trying to homogenize the brain regions properly. But when we look at the more summary of that, we see that there's clear indications of subfunctionalization, where the expression is kind of reciprocally retained in the 2 copies of zebrafish compared to the GAR. So basically the 2 genes, if you overlay the expression, resemble the expression in the GAR. And we also see regions in the teleos brain where we don't see expression in the GAR. And we're now following up also on this with HCR, so where we can multiplex both copies in the teleos together to see that it's actually the same cells that are expressing these paralogs. But of course, nowadays everything is going single cell, and here we are following the great initial work from Max. So Max, in his postdoc, was one of the first to look at cross-species analysis of single cell transcriptomic data, and also actually looked at the question of how gene family evolution and how these teleos extra copies shake out in the zebrafish teleos compared to the cavefish, the cave population, and the surface fish in the hypothalamus. And he found interesting patterns of these duplicates being differentially expressed between zebrafish and the Mexican tetra. And those are relatively closely related to each other. So we already see, or he saw, some interesting differences between these relatively closely related species. What we want to do now is basically take the outgroup also into comparison to figure out when did these changes occur. So how a PhD student, a computational PhD student in the lab, benchmark all kinds of cross-species methods for single-cell analyses. It's a rapidly evolving field, but the cross-species comparisons are still quite challenging. So there's a new paper coming out in Nature Methods soon. You can read the preprint here, where you compared all kinds of, not only for single-cell analysis, but all kinds of cross-species computational approaches. And we are trying to use methods now that generate metagenes or even metacells to do these comparisons across species. So together with Jamili then, we have generated data for the GAR brain, integrated it with existing data for the zebrafish using the Saturn pipeline, and then cell annotation with Jeff Farrell's Daniel cell atlas. And they overall quite nicely integrate. So the greens are here all the integrated cells between the 2 species. We see zebrafish specific ones, we see some GAR specific ones, which is probably due to differences in how the brains were prepped. We also do regional integration so that we can compare also the different regions of the brain across species. And then we are looking out for these patterns of subfunctionalization and neofunctionalization. So with subfunctionalization, again, GAR is expressed in these two regions here, and then the zebrafish homologs have become more specialized for them. Or we see some new or expanded expression for one or the other zebrafish duplicate, which could be indicative of neofunctionalization. So here just 2 examples. A glutamate transporter that shows this expected pattern of subfunctionalization, SLC17A6, where the B and the A copies seem to together, at least for some aspect, resemble the more ancestral, supposedly ancestral expression pattern that we see in GAR. And then here's some neofunctionalization where we see expression of the male to B duplicate that we haven't seen in GAR. So this is ongoing, and we're also now trying to go beyond the regional comparisons and go directly at cell types for themselves. And eventually, as I told you, we just don't want to do it with GAR versus zebrafish. What we want to cover, like, what is the potential of these extra copies across teleos diversity. So we will do this in comparison to medaka, to eels, and to butterfly fish, because they they represent the 3 main lineages of the teleos fishes: the bony tongues, the elopomorphs, and the clupiocephalans. There we will cover basically the entire diversity of teleosts. And we'll see whether what we see in zebrafish has played out in similar ways in these other lineages or in very different ways when it comes to sub and neo-functionalization. And finally, I want to give you some overview of what we do at the functional level for GAR. So we have worked with many different groups over the years to make the GAR a more accessible model for comparative vertebrate biology and work with the experts of different morphological structures in vertebrates. And overall, what we can say is whenever we include the GAR, it becomes, or the holosteins, it becomes obvious that many of the traits that were thought to be teleospecific or tetrapod-specific, they actually are representative in GAR and thereby were already present in the bony vertebrate ancestor. So for example, when we look at the connections of the brain to the eye, in humans we have contralateral but also ipsilateral connections, right, which gives us our bilateral vision. In teleosts, you only see the contralateral connections, but in the GAR system, we actually see something that resembles what we see in tetrapods. So again, it's not a tetrapod innovation that has something to do with vertebrates coming onto land. GARs also have a very remarkable regenerative potential. When you chop up an entire pec fin, it will come back after a couple of weeks, something that even zebrafish usually doesn't do. So how can we then improve on this? So Brad Rosico, my former fish manager, did a fantastic job to improve the spawning and husbandry methods for GAR. And then we're also trying to establish functional genomics, transgenics, and gene knockouts. Here's the first proof of concept of CRISPR genome editing in the spotted GAR. So we knocked out genes involved in melanin synthesis, and it works quite nicely. So you can get albino GARs by knocking out tyrosinase or OCA2 in a transient manner. And this is a later stage of an OCA4 fish. That's a mosaic, but we're now also getting better to grow up these fish and hopefully do some more functional work in the future with them. And this is kind of my vision for the future. So our vision for the future, I work with Zhanrong Wang at Michigan State. He is an expert on chromatin architecture, and he has developed methods for single cell high C of individual cells. So he has this 3D method now. This is chromosome 1 in the human genome to look at the folding and the interactions. And we are just starting to develop data also for GAR for this. So that's like 3D genomics that we want to do, and then adopting methods from our collaborator Alain Chedodal in France, like this iDisco tissue clearing and staining to then do also 3D phenomics in GAR and also zebrafish, so that we can then see where these genes are expressed in the GAR brain or in the GAR body and compare that to the 2 copies in the zebrafish and other teleosts. So hopefully in the future we can integrate 3D genomics and 3D phenomics to come to a more holistic understanding of vertebrate biology. So I hope I could convince you that the holosteins are really informative to learn something about genome evolution, biological phenotypic evolution, and then I've derived from them also something about disease phenotypes in vertebrates. And with that, I thank all the people that are involved in this. So it's been a great pleasure to establish and work in the spotted GAR and bovine genome consortia, and now doing this in a much larger fashion with the people of the VGP. Our funding resources, John, of course, with whom I could start this, and then my own lab at Michigan State, present and former members. And I'll leave you with this image here from René Magritte, which I think shows us that, well, we are just fish with limbs. And I don't know if that's a coincidence, but I want to believe that this is the beagle and shows the evolutionary component of this. And with that, I'm happy to take any question. Thank you.
Questions?
record was so rich with diversity. So does that suggest that maybe this idea we have that teleos diversity is all because of that whole genome duplication is not the whole story, or?
Yeah, so I think it's not really clear why the Holocene crashed and then the teleos came up. So of course there were also mass extinction events that contributed to that, and it took quite a while until the teleos actually took advantage of the genome duplication. So there's a gap of several tens of millions of years. So at first there was a lot of criticism that the genome duplication has nothing to do with it, because we don't see like 5 minutes after the genome duplication we see all this speciation happening. But the literature is much more rich for genome duplication research or polyploidization in plants, because they duplicate all the time, right? And that shows that there's also usually a time lag between the duplication and innovations or diversification, and we think that is going on here as well. So that doesn't tell us why the Holocenes crashed, but they are not as competitive to go in all these different potential niches. They're like tanks. They have this heavy armor. They're not the best swimmers, so to speak. So I think over time it played out that the teleos had way more potential for evolvability.
So you talked about how you can use this Holocene bridge approach to compare the regulatory regions between tetrapods and teleos. Do you think you could also use it to compare non-coding regulatory regions between different teleos species themselves, because their sequences evolve so fast?
Yeah, so so there are no real slow evolving teleos as far as we know, but when you want to go from these distant lineages like the eels to zebrafish, let's say, instead of trying to go through directly, it's actually better to go to Gar or to the Holocenes, find it there, and then go to the other side. The interesting aspect is also that, as similar to what happens to the genes, the regulatory elements after duplication don't get conserved in the same manner in these deeply diverging lineages. So sometimes it might even not be possible to find the elements, or you find them on the different duplicate. But that's something only now we can look into, because we have these higher quality level genomes for these early branching lineages. Before they were only crappy scaffolded genome assemblies, and now with the VGP and other ways we have the genomes to do those comparisons.
Is it known, or can you speculate why or how some lineages maintain all those mini chromosomes and other ones?
The microchromosomes,
Microchromosomes.
Yeah.
Versus fusion.
Right. So I think it's an illusion to think that microchromosomes are something unique. So the more you look into the karyotypes of vertebrates, you find microchromosomes basically everywhere. And some lineages have fused them. Microchromosomes have some special characteristics, like they're very gene-rich, GC-rich. They are, according to 3D genome reconstructions, organized or like interacting a lot with each other compared to the macrochromosomes. But we don't have a good view yet what that actually all means. The fusions, because with the comparison between Gar and chicken, we can figure out which are the old microchromosomes and then trace them into the different lineages that don't have microchromosomes, but it's not always the same microchromosome fusions. So at this point, we actually don't know. Yeah.
So you mentioned you use the ataxy for the developmental patterns for the teleos and the Holocenes. So I was wondering if there were any notable differences between how the developmental patterns are different between these two.
Yeah. We don't. Again, we don't see any dramatic differences. The main two differences we see, of course, is A, with the holosteans, we see more conservation over to the tetrapods, or more readily conserved. And then B, we see this pattern of looking at the duplicated regions in zebrafish or in the darker. So I think what we will see once we have done this all and properly integrated all available data is that there is a trend that when you look at the two copies, that one of the two copies keeps a lot of the ancestral elements, and thereby also open chromatin regions, and then the other one, relatively few and also fewer that will be open. So there were some indications early on when people just looked at sequence conservation and they called it like winner takes it all or almost all, and I think that's something we're gonna see. That has also been to some degree seen when people did ataxic and amphioxus, like in chordate, non-vertebrate chordate, compared that to chordate to vertebrate data. The problem there is that you don't see sequence conservation because it's so divergent. You can only count like the open chromatin regions between the single copy and the multiple copies. I think here we have the chance not only look at the open chromatin regions and count them, but also look at the sequence conservation of them.
Thanks for the great talk. It's kind of a broad question, but I was wondering if there's some sort of like mechanistic trait across some of these like living fossil groups, like turtles and like holosteans, where what is it about their genomes that makes them like slow?
Yeah, that's also a very good question for which we have no satisfying answer yet. So there's multiple aspects that play into sequence evolutionary rates, metabolic rates, generation time. So, I mean, the holosteans are kind of having long generation times. They are having maybe overall reduced metabolism compared to other lineages. But other lineages that show similar characteristics don't necessarily have slow evolving rate. So there have been some suggestions that they might have a particularly good DNA repair control. So that's something we're currently looking in. So we made crosses between, normally we do these crosses, these spawns as group spawns, but we were successful at getting like single pairs, and then we have the offspring. So now we're sequencing the offspring and the parents to figure out the germline mutation rate in GAR, and there's a lot of data from across vertebrates to see if there's maybe a slower mutation rate or lower mutation rate. But again, that wouldn't necessarily explain the mechanism. So we still need to do a lot to figure out what's going on there. Like the sturgeons are really confusing to me because they went through these many different rounds of genome duplication. And we always take genome duplication as our explanation for why teleosts are so fast, but then the sturgeons have a relatively slow rate of sequence evolution as well. So yeah, it's an open question.
Maybe in terms of time, we can say, thank you for the talk.
So illustration is so many regular regions. So what is it that you're identifying? Is it the same like transcription factor binding motifs? Or are you identifying, like, or using some kind of like bridging method where like the GAR shares something with humans, they share some binding motif, and then it's a different binding motif they share, or is it just like? Do you know what I mean?
It's a mix. So one thing that we found, which is quite interesting, is that very often we see, we have a sequence conservation block from GAR to human like this, and then from GAR to zebrafish like this. Is this the angle that's like in the TVXT case, yes. So they're kind of overlapping, but the kernel of the conservation is overlapping.
Yeah, of course.
So it could be that there's even some sub-functions within.
Yeah, that's
Then they kind of diverge. Like also in the TBXT, then ZBRF or the TBS had like another TBX binding motif over here that was within the TBS well concerned.
It's also
The
looks
remote
really
didn't
good.
have. So
So it's kind of like, is that how you're kind of thinking about it, like sub-functionalization?
yeah, like at the level of the element potentially as well. Maybe sub and neo functionalization.
And so rather than like evolution of new models.
I mean, those of course are also happening, right? So like, talking with Mike, I mean, we have a pretty
Do you want to pack up?
strict strict definition, but of course you could also say, oh, if they are within 200 kb or so of each other, you could call them they're in the right region.
Yeah.
So, but yeah, it's a tricky business to call the
Whether it's
kind of
more-
arbitrary sometimes to how you filter it.